We are super excited to be here. So without further ado, I'll introduce our speaker tonight. Dr. Michael E. von Fricken is an assistant professor of epidemiology in the Department of Global and Community Health. His research interests include vector-borne disease surveillance, control, and pathogen discovery. He has ongoing projects in Kenya, Mongolia, and Haiti, focusing specifically on emerging pathogens transmitted by mosquitoes and ticks, and has conducted international training workshops on appropriate field methodology and data management for vector-borne disease research. So his work has a direct impact guiding treatment policies of rickettsial infections in Mongolia and his management of malaria in Haiti. He received his PhD from the University of Florida and completed his postdoc fellowship with Duke University, Divisions of Infectious Disease. He is now currently a research associate with the Smithsonian Institute, National Zoological Park, and holds a visiting scientist designation with the U.S. Army Medical Research Institute of Infectious Disease Diagnostic System Division, based out of Fort Detrick, Maryland. So without further ado, thank you so much, Dr. von Fricken, for joining us tonight. Thank you so much, and go ahead.
Okay, great. Thank you.
Thank you. You want the...
Can I use both?
Yeah, absolutely. You can if you want.
So this isn't a camera, right?
Nope, it's a speaker.
Oh, thank God. For everyone who's watching, I'm very handsome. Well, thank you very much for letting me join you guys today. I'm going to talk about a One Health approach to investigating vector-borne diseases. And so this is what I've been doing through my postdoc at Duke University and on to my research career as an assistant professor at George Mason. And just a quick disclaimer, because you never know who's watching these things. The information in this presentation does not reflect the stance of George Mason or any of my collaborating partners. This is our findings and my personal interpretations as well. So I've already had my introduction, so I don't know if I need to go through this again. But that was when I used to be a lot skinnier and living in Haiti. One of the downsides of going into academia, I suppose, is a wider waistline. And I thought we could just jump right into our work in Mongolia. The sites that are highlighted are our sampling locations, the SOMs that we were working in. And the purpose of our research was to address tick-borne diseases using a One Health approach. And of course, most of you know what One Health is, but it is the discipline of bringing in expertise for medical, veterinary, and environmental health, and looking at the interplay and the connectedness of how diseases and chronic conditions can translate across the disciplines. So for this study, our medical health was looking at high-risk herder populations and finding out their disease status, as well as administering behavioral surveys to figure out what kind of exposures that they might have to tick-borne diseases. Our veterinary health was, of course, sampling goats, sheep, cattle, horses, camels, and rodents across Mongolia, as well as administering household surveys looking at animal health, and then collecting ticks off of livestock and rodents. And then, of course, our environmental health was sampling ticks from the environment, as well as looking at vegetation indices, land surface temperatures, and other remote sensing data to see if we can find information about the ecological niche of these tick species. And of course, I've got lots of pictures, and I'll try not to bore you too much. I'll read the room. In our research, we had doctors, veterinarians, mammalian specialists, epidemiologists, environmental scientists, local herders, microbiologists, geneticists, and of course, government partners from the National Center for Zoonotic Disease, the Institute of Veterinary Medicine, and the National Center for Communicable Disease, just to name a few. So there's a lot of different people that go into a large-scale project that you have to play nice with. And that's one of the things that you guys, if you start doing One Health research, you'll find out quickly, is this an approach or is this a mandate? You know, sometimes you have trouble bringing the right people into the same room, and will they even communicate? And so you have to be able to play within the politics and make sure that partners are communicating and have the same shared vision of addressing the common goal. And of course, this is our D43 Team 1 researchers. That was funded by a grant to Dr. Greg Gray from Duke University to investigate tick-borne diseases.
Just gonna make sure it's showing. Sorry.
Oh, it's okay.
Oh, excuse me. All right.
So how's everyone doing? my opportunity to practice my stand-up career. And so what we'll be looking at is how to answer these questions using the One Health approach. What is the distribution of ticks in sampled regions? What pathogens do they carry? What are the potential reservoirs of tick-borne diseases? and then our herders and their livestock actually being exposed to these diseases. If you're going to find diseases in ticks, but not in the population of interest, is that a major concern? You need to make sure that there's their interconnect, that people are actually being exposed to these diseases. Are there specific behaviors that put people at an increased risk of exposure? Is there a way to forecast the risk of disease exposure? So this is a big push in One Health and in global health and in research for infectious diseases. You take all of that lab data and every single person you've sampled and surveyed, how can you take a subset of the population and apply it to a larger region? How can we forecast? How can we predict? How can this small sample tell us about the greater population? And then what else is missing? So where we worked in very distinct geographic locations. We worked in Seling with Tushuk, Erun, and then in Tov, in Tillich, and Dongobi, Sanchand, and Taranjalan. My Mongolian is terrible, so if anyone from Mongolia is listening, I apologize. And what we have is the high mountains for northern Mongolia. That's about above 1,600 meters. The central steppe, and then of course the Gobi Desert. Anyone here been to the Gobi? Keep it that way. It's not very pleasant. But each spot had very unique environments and different tick species that we were finding. And so with different tick species, you're going to find different diseases. And of course, you have to consider the populations that you're working with. You have a nomadic oral history of illness. It's good to listen. You have the black Saxolon bush or the Saxo bush, and you have the Siberian birch. Both of these trees are known as illness trees. And they've been known as illness trees for over 1,200 years. They don't understand that it's because you have Ixodes persiculatus, main habitat in the birch forested area, and you have Hyalomma, asiaticum near the Saxo bush. All they know is that when you spend too much time where you live close to these trees, you become ill. And so there's sometimes stories that can tell you a lot more about infectious diseases. You just have to listen. And this is what I did for a year. It was a lot of work. We enrolled 400 nomadic herders with GPS points, had surveys on a majority of them, almost 900 goat, sheep, and then you guys can look at the numbers. Basically, we looked at a lot of different livestock and the herders that own them. And we were trying to see if the livestock could actually be a sentinel for disease status and disease exposure in the herders, so that you don't have to go in and test every single herder. Maybe you could go into a community and screen X amount of goat or X amount of sheep and figure out how much of a disease risk is this area based on what you're finding in livestock. That didn't work so well for us because exposure was ubiquitous. There were so much tick-borne diseases in the population that it was very difficult to find enough variability to find an association for a sentinel population. Still important. And of course, we have our livestock samples. Being one of the larger guys on the team, I had to literally take the bull by the horn on many occasions. That was one thing I was good at, I suppose. And we were trying to see if livestock could be used to be a sentinel. So moving on to what ticks we're finding, and this is an overlay of NDVI, or normalized difference vegetation index, and it tells you how much light is being reflected off of green forest or grassland versus arid regions. So the areas in green are going to have more forest and grass coverage. The areas in blue will be arid, flat areas of the Gobi. And we'll see 3 different genus of ticks. Demersen natali were found throughout Mongolia. Hyalomma ticks were only found in the Gobi. And of course, our Ixodes persiculatus ticks were found in the forested birch regions in the north. And this matters, because each of these ticks transmit different sets of pathogens. And so if you're looking for a certain disease, you should know your enemy. You should know where you're looking. And you can actually use vegetation index to actually, guess isn't the right word, but take an educated guess of what you might find in certain regions and certain latitudes. And one of the more interesting aspects of collecting Hyalomma ticks is they are active host seekers. So have any of you guys collected ticks before? I know Heather has. Some of my students are raising their hands. It's not fun. Collecting Hyalomma ticks is even less fun because you don't actually drag. You stamp around and they sense the vibrations. So you are essentially the bait as you're stomping heavily through close to these bushes to hope that ticks will come out and chase you. Yeah, and so for a majority of bacterial infections, a tick needs to be attached for about 24 to 48 hours. So it's okay, you can do the checks, We actually had to suit up in your Tyvek suit because these viruses can be transmitted in less than 30 minutes. And the number keeps getting lower and lower, and I'm like, don't tell me this. This is too scary. And I always had this fear that when we're doing research on new emerging pathogens, that I would be this point source of an infection from the middle of a goby where no one's been, and I get some tick bite, and you bring it home. And that's a very real fear. So you have to be precautious when you're doing this research. You will be at the front line of One Health research. You might be collecting bats, rodents for arenaviruses. You have to make sure that you aren't going to be catching the one thing you're trying to prevent.
But if it's a bacterial, you can have it named after.
Well, there's a lot of those these days, especially for Rickettsia. Stop naming new ones. They're not that different. But I will have one day. And so we figured out a general idea of what the distribution of ticks are in the sampled region. Now it's time to look at what pathogens these ticks carry. And there's a couple different ways you can do it, but essentially you're going to be looking at PCR and sequencing. One of these was published in 2017, and we have another one under review, looking at Rickettsia, Anaplasma, and Ehrlichia. And anytime you can add sequence data to your research, that's a major plus, but you have to know what you're doing. You have to be targeting the right gene fragments, where you can tell that you're looking at a different species. Otherwise, you're going to get confused and misidentification. So a good rule of thumb is target multiple genes, look at the literature, and say, okay, what does this say to GenBank? Is this enough variation? Is this a new identity? And the second you think you have a new pathogen, test it against some more genes. Because you don't want to come out and say you've got something different when it isn't. And the other benefit of doing sequencing is you don't always know what the clinical isolates are in the region of interest. But you can understand what's happening in the neighboring countries. So we know a lot of people are dying of tick-borne diseases in Russia and northern China. And they're sequencing those isolates. Well, now we can take what we're finding in ticks and compare that sequence and say, okay, this is 99% similar to what has a 10% fatality rate in cases that are reporting at hospitals. I can put a safe guess that this is probably pathogenic in humans. And so it's just one more tool in your One Health toolkit of looking at infectious diseases that are emerging. And so from our Rickettsia work, we found that about 15 to 30% of the dermocentric ticks were positive for Rickettsia ralti. And then more interestingly was 46% of Ixodes persilicoides ticks have a relatively new Rickettsia tercivich, but only one positive pool for tick-borne encephalitis. And I'm going to explain why that's important. Oh, and here's our wonderful phylogenetic trees. Next to our Rickettsia tercivich that's bold, that's a human case. Again, for 2 target genes, and then all Rickettsia raltis are also human cases. So we're trying to find, are these pathogens actually going to cause clinical symptoms in humans? So back to the tick-borne encephalitis. Why is this important? Well, for Rickettsia tercivich, it's atypical for spotted fever group infections. Usually you have a rash, it's spiked fever that you'll see with other Rickettsias like Rocky Mountain spotted fever. Instead, for Rickettsia tercivich, what they were seeing was neurological symptoms. Well, the other endemic disease in northern Mongolia is tick-borne encephalitis. They had 4 case fatalities of tick-borne encephalitis, none of which were confirmed, but they said this is tick-borne encephalitis, palliative care, and see if they recover. Because of this research, because of this simple finding, now there's a new change in policy that anyone who has tick-borne encephalitis is actually going to receive antibiotic therapy as well to make sure that it's not a misdiagnosis for Rickettsia tercivich. So there are real policy implications to your research, and it feels great when you get it right. And our next step is actually to look at all the samples we have and see if there's any human or animal isolates for Rickettsia tercivich. Our Anaplasma work, we found lots of Anaplasma ovis. This is responsible for $70 million in economic loss in northern China every single year. About 15% of the ticks tested positive. Have any of you removed ticks off of livestock? Are there usually more than one? How many do you need to have attached to it to get an infection to occur here? And so if 33% of your population is nomadic and persists on livestock, and this is a major part of your economy, you have Anaplasma ovis is doing damage to these herds, spontaneous abortion. They can cause anaplasmosis and kidney failure in sheep and goat. We should be taking a look at this, especially when it's their bread and butter for their economy, sheep and goat. And this is just one more component of necessary tick-borne disease research. We need to look at the holistic profile of diseases that are endemic in Mongolia. Here's some more trees. If anyone's interested, I don't know if this is the right, well, probably. I think you guys are following. Essentially, the closer they are, the more similar to other isolates that have been sequenced. So anything that says Mongolia tick is one of our ticks, and they're looking at the ticks in the region. So we know what some of the pathogens the ticks are carrying. We haven't looked at Borrelia. We haven't looked at Crimean Congo yet. There are a lot more diseases that we're gearing up to test, but we're starting. The other thing we decided to look at was possible reservoirs of tick-borne diseases. If you can't figure out where all the ticks are, you can find out potential reservoirs, and there's a lot of detail on mammalian populations and rodent populations in Mongolia, where the distribution is. If you can identify 2 or 3 possible reservoirs, that might also tell you something about disease distribution. And so it's one more aspect of a One Health approach. And this was Laura Pulscher's thesis project. She came out to Mongolia and was working with us. And one of the key findings she found was the Mongolian gerbil had Rickettsia DNA in 17 out of 18 samples tested. So that's a big clear sign. I mean, even though it's not the largest sample size, having 17 out of 18 gives you some confidence about this is a possible or very likely reservoir of disease. And of course, we also found that other reservoirs can carry the diseases of Rickettsia and Anaplasma as well. And so that complicates it even further. Here in Virginia, our white-footed mice are very competent vectors of Borrelia, and we're finding more and more out about chipmunks and other rodents that can carry Borrelia. That's one more reservoir of disease for ticks to attach to and have infection at an early stage, in the larval stage. So, I know, I'm like hammering you on the head. I'm forcing everyone to read my research. This is the wonderful part of the captive audience. No one can escape. Lock the doors. We're now moving on to our herders and their livestock actually exposed to these diseases. We know they're in the ticks. We know they're in the rodents. And we know that they have a pretty wide distribution. The answer is yes. I know everyone's on the edge of their seat. What we're looking at are using IFAs to determine previous exposure to Rickettsia and Anaplasma. These pictures aren't that great because they're taken on my Mongolian laptop outside the lab, but they work. You'll see a starry night pattern with Rickettsia, although we can only see one star, and we have inclusion antibodies inside for Anaplasma. And this figure will show that, remember I wanted to talk about sentinel populations and maybe one will predict it or not? It did it. All the animals had some levels of exposure. All the humans had some levels of exposure. So there wasn't that, we figured it out, we cracked the code, we only have to look at sheep. It's not that simple. This is much more complicated. But this is a snapshot of exposures in nomadic herds. There are things to be learned by seroprevalence studies. What would you take from this information? I'm actually asking you a question, you guys. We can be interactive. I know the webinar people are like, what do you think? All right, I'll go.
I'd rather go hiking down.
Well, that's the Gobi. You don't want to go hiking down there anyways for other reasons, but you're right. There's a lot less tick-borne disease being transmitted in the Gobi region because that's not as suitable a habitat for ticks. But as we're moving closer and closer to the north, we're finding very high seroprevalence rates of Rickettsia exposure and Anaplasma exposure. And so if you're targeting research or you're getting your next steps in order for additional research, you can say, let's stay above the 48th latitude. Let's go up. Let's see what else is there. And that's exactly what we're doing next. And then, of course, we have our survey. And this was, it was tough to actually administer a survey for every single participant, because you have to recall we're enrolling about 100 livestock from each herder, and then taking blood from every herder, then administering a survey, and winter is coming, literally winter is coming, and so you have about 3 weeks to do all of this until you start getting sub-zero temperatures. But this actually was very interesting. We found that there was major disparities between tick bite recall and tick bite exposure, and that happens here in the US. But we had a subset that said 64 had said they'd been bitten, 34 had said they had very severe symptoms of rashes or severe fever or SR. Only 2% of men sought treatment and 5% of women sought treatment. So of the 100% of individuals who could have received treatment, who had symptoms, 2%, 5%. These are nomadic herders. They are very far from hospitals. They're extremely tough. They have constant aches and pains, and they assume it's the weather, and that's it. But you're missing a population here. We aren't providing the proper medical treatment. Other information that we discovered is the reported tick bite months. So those are going to be the months that I'm going to be in Mongolia in the spring. You want to target your time, I suppose, to the peak seasons. And then, of course, is it possible to forecast this? This is where it gets a little more confusing. You want to take all of the data from your seroprevalence study, your tick detection rates, and put it into an agent-based model. It's going to simulate potential exposures moving through time. And so we have our sheep, our little white dots, the blue humans, and then I don't think you can see it from there, but there's lots of little dots of ticks. And as the model is being simulated, all of them are moving through different environments. Different environments have different infection rates, and there's a probability of getting a tick bite or not. We ran this about 5,000 times for each model, and I'll break it down quickly. So if you're in a high forested region, over your lifetime, you're likely to have 16 infectious tick bite exposures in your lifetime. This is only based on 2 tick-borne diseases that we've included in the parameters. We haven't included any Borrelia, which is probably even higher. We haven't included Rickettsia or tick-borne encephalitis. There's other diseases that could go into here. And so that number is only going to go up. If you're spending 8 to 12 hours herding livestock through endemic tick areas, you're going to get tick bites. Even when we are surveying an individual, I've never had a tick bite. I never had a tick bite. His wife, right here. Okay, he had 1 tick on his neck. We removed it. Okay, I've had 1 tick bite. This is the population you're dealing with. This is not a concern. They don't view it as a concern. And that means you have to have an education component. This can't be colonial research. You have to come in and actually have an intervention and provide this information to affect policy, to empower populations to take their health into their own hands and make sure that they can receive proper treatment. And so what next? We have a trip to return to Mongolia spring 2019 and expand our tick-borne disease research. Our last trip, we did a vertical stretch, and this time we're casting a wider net and really going to look deeply into the tick-borne diseases that are carried. We're going to use multiplex assays to identify multiple pathogens in a single run, and then next year, targeted human surveillance, targeted livestock surveillance, to really paint a picture of what kind of exposures are happening and what is the clinical epidemiology of these diseases. Plus, we can plug more things into our pretty model. This is our sampling approach. So this is 24 days in Mongolia, and that is the equivalent to about 4,000 kilometers over 24 days of camping. I'm bringing 4 people with me, so some of you guys are saying you're looking for opportunity. I don't know if you want to be punished with this, but we're going out and we're really looking hard, because right here we have Haemophilus genus ticks. We have severe fever, thrombocytopenia syndrome that's occurring in Asia. We now have the Asian longhorn tick here in the US. If we can start looking for isolates in Mongolia, we can actually get those to the US or to military bases that can start taking apart and picking apart the sequence, looking at specific proteins and figuring out potential treatments or other novel diagnostic techniques. Right now, we don't have as many isolates of it as we should, considering that this tick is in our backyard. And so that's what this big push in the center is, because that's a known environment that we missed in our first attempt. And then, of course, we're going back north. Luckily, I got to pick the trip, so I was like, no more Gobi. No more Gobi. I'm done with the Gobi. And so you take this all together, the One Health approach, and tick-borne diseases are a very real emerging threat. I don't know if emerging is the right word, because I think they've just always been there. And people die of strange diseases, and they say, oh, well, the temperature dropped to -50, it's a tough life. But it's there. So what we need is improved surveillance and response. desperately. We need to start getting out and engaging our governments in Mongolia to have their satellite branches reach out to herders and communities about these diseases, about treatments, about testing, and to actually maybe have them start doing tick checks. I don't know if you could do enough tick checks. It's like you're almost in the battle zone all the time. You have to get fatigued. And so maybe we have to have them be aware that if they have an illness or they're feeling general fatigue, that it isn't always aging, that they could have some other disease that's causing this. And finally, using the One Health approach requires shared vision across partners. You really need to have everyone on board, and you can't have the infighting that does happen sometimes. And if done correctly, you can look at, investigate hypotheses from multiple perspectives and use data that might not be used the same way in the siloed approach. If it was just me, I would have only looked at the ticks, because that's what I would have been interested in. But now I'm looking at the herders, I'm looking at survey data, and trying to bring it into an entire holistic approach. And I think it paints a much better picture of tick-borne disease risk in Mongolia than, look at all these sequences. I'm done. You know, One Health is about bringing people together and having a common goal. One of the downsides is you end up having about 15 to 20 authors on the paper, but that's okay. You want to have everyone together and addressing these issues, especially large-scale projects. And of course, our acknowledgements. This is not just, you know, the My Fun Fricken show. I'm one small component of a lot of individuals who put blood, sweat, and tears into this project. Our D43 team members, Bulbatar, Batat7, Halak7, Supatra. They were the PhDs and veterinarians from Mongolia I worked with. Very, very wonderful people. And of course, we have, this would definitely not be possible without the financial support from NIH to Dr. Greg Gray to investigate zoonotic diseases in Mongolia. And with that, I am open to questions. Thank you very much. Yeah, sure.
Before we start questions, I would like to present Dr. John Fricken with the usual gift we provide our speakers. So thank you so much for coming. This is a flag that has been flown over the Capitol.
Oh,
so please
wow.
Enjoy.
Thank you.
Sorry for all the parking issues and communication struggles.
Worth
but
it.
Thank you for putting up with that.
No, thank you. That's very
and
nice.
coming to share all this awesome information with us. So all I'll ask is that if you don't mind repeating into the speaker the questions that are asked of you.
I'm going to struggle with that, but okay, I'll try.
Well, I'll help out. But yeah, any questions?
Thank you. Really enjoyed your presentation. One of the, back in the early, or in the mid 90s when the whole emerging disease issue started, the title was Emerging and Re-Emerging Diseases. But I don't think that made a very good acronym, so they threw away the re-emerging piece. But as you say, the ticks and perhaps these diseases are re-emergent from our perspective. My question is, a lot of times, especially with vector-borne diseases, it's not only the behavior of the vector, but the behavior of the individuals. What puts them in harm's way, if you can call it that way. Did you have any sociological aspect of your research? And if not, can you overlay that the next time that you're there to look at what behavior is placing them in, proximity of the tick?
Sure. Do I need to repeat that? So the gentleman first said he very much enjoyed my lecture, and very, very, very much, and was asking about social and behavioral factors that could put an individual at higher risk of tick-borne disease exposure. We did have some of that, and one of the variables that was significant was the amount of hours per day spent herding. But once you got above 6 hours per day, everyone was pretty much exposed. It's tough to find variation with this population because they live, 6 to 8 people in a gur that's smaller than this entire room. Their entire livelihood is their livestock, either through milking cattle or grazing sheep and goats. And horses play a major role as well. So when you're riding a horse through the forest, it comes up the horse's legs and eventually attaches to you as it's going upwards. So it would be tough. I would think surveys should actually be targeted looking at health-seeking behaviors, since we have a good idea of the risks of tick-borne exposure. I mean, you can't get these guys to wear DEET. I guess you could use paramethrin-treated DELs that cover their whole body. That's their traditional garb. But it would be better, I think, to look at health-seeking and figuring out unique ways of increasing access and point-of-care testing. So making sure that if someone comes in, physicians out there might say, oh, you've been eating too much meat, so stop eating meat and take these vitamins and come back in a few days if you're still feeling sick. If you can easily test someone for multiple pathogens right then and there, doxycycline will take care of most of them. I mean, and I'm not advocating saying to give doxycycline to every individual that comes in, but if they have a history of tick bite, absolutely. There's just too many that are being transmitted. And I sometimes have conversations about Lyme disease here in the US and the terror and fear. Like, oh, we don't even have Crimean Congo, guys. Like, this is a treatable bacterial infection there. They've got stuff that can kill you. And so I think looking at those social behavioral determinants of health seeking care might be a little more interesting, because we know that they're just having tons of exposure from being out in the environment. Yeah, of course.
I don't think I've ever heard, how do you go about getting the consent for the herders to participate for them? And how do you go about explaining it? I'm curious about that.
If anyone's done any human research, you have to have IRB approval, and then we have Ministry of Health or the Ministry of Sport and Health in Mongolia, where they see our informed consent form, and then they look and make sure that there's no coercion or undue influence, and that there's no unnecessary risk. So those institutes are in place to protect participants, not us. They don't care if I get a tick-borne disease out there. They're interested in make sure that I'm handling our participants and their information properly. And so before we can even do anything with their animals or the participants and the herders, we have to have full permission through IRB and IACUC to work and take samples from them. And at any point, they can contact us and say, destroy that sample. And I have to go, figure out a way to get to Mongolia to toss that one sample and document it. No one's done that yet, thank God. But you have to protect your participants. So this is voluntary research, and one of the things that we struggled with was getting this information back to herders, and that's something we really couldn't do because they're nomadic. They have 3 or 4 spots that they'll stay at, but what value is it to come out and say, at one point in your life within the last 5 years, you've been exposed to a tick-borne disease? And they're like, oh, no, that's not me. We're here in this grazing area. That's someone else next year. It's really tough to track these individuals. They don't have phones, and they move. And so what we said is we're going to take this information and provide it to your SOM centers and your SOM leaders and provide training to guide the overall policy and our understanding of tick-borne diseases and their epidemiology in your local region. And they said, okay. And I mean, it's a great honor to let us draw samples from their livestock, especially their horses. Horses are sacred in Mongolia, and to handle them, to be able to draw 287 samples from horses, especially the horses that they're racing, I mean, that's very generous, to say the least. So.
So I think you touched on it a little bit earlier in the presentation, but I was wondering if there was any effort either in the past or like as you're moving forward to this next project to incorporate any amount of traditional knowledge from the herders and like kind of seeing what solutions they might have come up with to these sorts of problems before. I mean, you talked about how they know to stay away from certain plants. So like what other aspects of that have you been able to come across or incorporate into preventative medicine?
Sure. So this is, do I have to repeat the question? Yeah. Okay, I'll try. And so I was asked if there are future efforts planned to look at cultural beliefs and norms about tick-borne disease and tick-borne practices of health seeking. So the cultural side is actually a two-edged sword. They have the plus side of knowing not to go by certain bushes and trees, and then they have a much larger negative side of seeking traditional medicine and shamanism, where it's praying, it's Hinduism, it's Buddhism, and it's reflection and fasting and other things that don't actually quickly treat the bacterial infection. And so there are risks, actually, that I would say that are more likely you're going to find more risks in traditional medicine as far as disease progression and our understanding of the disease. But I think that's also important when you're in these homes, that you have to listen. And so it would be great to do unstructured focus groups and interviews and allow them just to talk. You pose a question and let them speak for 5 minutes, and then you find one of the 5 million people who speak Mongolian on this planet and they translate it. It's a difficult language. And so I think that's something that we're missing. But again, I mean, we're just beginning our research here. I planted this question. Go ahead.
So I think more than like 50% or about 50% of the population lives in Ulaanbaatar. Am I remembering that right? So I was just curious if any of your research covered tick-borne pathogens kind of more in the urban center and what you might have found there, because there are certainly animals within the urban perimeter as well. I was just curious if there was anything notable.
Sure. So I don't think I was asked if I did any research on the capital Ulaanbaatar and tick-borne disease exposure in the capital region. I don't think there's actually any direct exposure happening in Ulaanbaatar, but a common practice is to go out to the countryside for a month at a time. Everyone tries to get out to the countryside at least 3 or 4 times a year, especially in the spring. You just made it through an incredibly harsh winter with high pollution. It's time to go back out into nature. People will just drive and camp. And I think the exposures are happening then. So working with the nomadic herder populations, we're able to kind of see the bubble around Ulaanbaatar where people are going to be exposed. So one of the regions we looked at is Tillage, the national park. I don't know if you went there or not. It's very nice. And that's where everyone tries to get to once the spring. And there's a lot of also religious beliefs surrounding it. And so you need to go outside and be with nature. Well, we were doing our part collecting ticks, making sure, let's see how bad diseases are here, because people then come back in to Ulaanbaatar with the diseases. It's much more common when they come from the north. They come in with rickettsioses or borreliosis, and they have actually really severe neuroborreliosis for the Borrelia variants they have in Mongolia. That's unpublished, but yeah. Yeah.
Congratulations on One Health Academy first picture in front of a monument with a tank on it. Excellent.
Thanks. Oh yeah, I got that. I didn't know I had the picture in there.
So where does the progression of perhaps seasonality go with other issues of climate change? Mm-hmm. And then what are the other borders? I mean, you're working within a political system in Mongolia, but what are the implications for other parts of that geography?
Sure. So I was just asked a very loaded question. So we had survey questions about climate change, and we had mixed responses. There were herders who viewed climate change actually favorably, but you have to imagine that they made it through enough -40 winters that if it's a -30 winter, that's a win in their book. But there were a lot of concerns about grazing lands and snow melts occurring too quickly and flooding, or we're not having the same amount of grass for herds. So climate change is an issue. You're going to see a proliferation of ticks moving south and north as climates continue to change. And Mongolia is in that little sensitive pocket between, Russia and China, to say those two interesting countries. And not to stoke, fears of anything, but there's so many unknowns for Mongolia. It's almost like Central Africa in Asia, because typically if there's a disease outbreak or a family dies, it's contained. The nearest person lives 15 kilometers away from them. They have anthrax, they have plague, a wide range of tick-borne diseases, as well as hantavirus, but they're separated. There's only 3 million people in the entire country. And one of the concerns is that we're having more and more trade across borders and more individuals congregating in the capital, that you could potentially have enough to sustain an outbreak of something unknown or something a little nastier that we haven't seen. During, the Black Plague was actually brought in from the Golden Horde into Europe. Mongolia was partly responsible for that. So if you have this spillover event into your neighboring countries, then it's no longer a small problem. It's no longer Mongolia's concern. It's the U.S.'s concern. It's China's. It's Russia's. And so having a good idea of what's happening in Mongolia is very important because Russia and China do a very good job of investigating emerging tick-borne diseases in their countries and other diseases as well.
You mentioned briefly that one of the ways you knew what to look for was that there was data from Russia and China.
Mm-hmm.
Is that unit coming from like similar populations of nomadic people, or is it coming from a set of people?
From urban populations and their general case reports that occur. And so someone shows up with rickettsioses, they'll have a full PCR and confirmatory testing, and then isolates get sequenced. Those sequences get submitted to GenBank, and then when we sequence our pathogens and ticks, we can match them up about what's happening in the clinical populations to the north and south.
Hi, thank you so much for your talk. I was curious, given the recent increases in cost of doxycycline in this country, if you'd encountered discussions about, in addition to the capacity and testing and education needs, about the cost of treatment for these communities?
You know, I actually don't know the cost of doxy there, but I would think the biggest issue is even availability of it, making sure, one, people know to prescribe it as well as take it, and to have it on stock or in stock. You can find it in Ulaanbaatar, no problem, but it's only in the I guess the equivalent of a state capital for Mongolia. You'll only find them there, not out in the outskirts or the satellite clinics, not all the time. So I think that's worth investigating and asking. But even just getting them to change policies of giving doxy more frequently, that's a start. Mm-hmm.
People tend to be more honest and maybe interested in things to do with their animals, especially if they're economic value in themselves.
Mm-hmm.
So the questions we're discussing were whether the person had ticks or the person had an illness. Did you ask the same thing about what they felt about their livestock?
Yeah, and they talked a lot about their livestock, and they had so many, so it wasn't possible to say this one livestock. And so they said, we've had brucellosis, we've had anthrax, we've had cardiovascular disease of animals dying of X, Y, and Z, but it was impossible to link that to a single animal with a serological test. And so that would be almost like an ecological study looking at the herder population that, I guess the livestock they own. For the individual survey, we were able to link that directly to any biomarkers of exposure. So we first looked at that, but I've got 81 surveys, which is, that's kind of small amount, but they're about 60 questions long about the health of their livestock, and they answered all of them. Yep.
Just a question that's been generating in my mind as a result of this discussion. Pyrethrum is a plant, mm-hmm. and we impregnate bed nets for malaria prevention, etc. You mentioned that they have sick trees.
Mm-hmm.
Was that the term?
Fever trees, similar to yellow fever.
Conversely, are there areas where they know are safe, and perhaps could the environmental health side of One Health start looking at, are there any local products that could be made to, I don't want to say impregnate their trousers or something like that, mm-hmm. but something that's practical that could be used to prevent tick bites.
Maybe. So they're almost fully covered, and it's called a dell. It goes down to their legs, and then they wear thick boots. I think they know more about where not to go. And then they have traditional grazing grounds that their ancestors and ancestors and ancestors used, and so they just continue to use those same properties, those same landscapes. I guess the downside to that is if there was a disease that was really deadly, natural selection would almost have that group possibly weaned out over talking about centuries of time. So I think it's more along the lines of knowing where not to go as opposed to where to go, if that makes sense. Yep, of course.
I'll wrap up with one final question. Oh. So the One Health approach in general,
what are some of the biggest challenges that you faced across all these experiences, especially with a global lens, that you came across that we might not necessarily know about or expect to face with your research and projects?
Sure. So I hope my mentor doesn't attack me for this. I think we actually need to move away a little bit from the One Health approach of we have our one veterinarian, our one environmental scientist, our one doctor, we have each person with us on file, and move more towards One Health thinking. So recognizing that you're out of your depth, and a sprinkle of some veterinary expertise would actually be really not just important but critical to the success of your project. That was one of the more difficult aspects of the study, is that we had every single portion in there, and looking back, I would have done things a little bit differently. And so the One Health approach is great, but we really need One Health thinking of just recognizing when our expertise is not enough, and looking at bringing in other people as a positive instead of as a competition or self-eating of our own grant funds. I think that would be my best advice.
Thank you so much again.
Thanks. Even though my presentation was on the One Health approach.
No worries. I will encourage everyone, just as we close up here, to network, and we'll be out of here by 8, so to talk to Dr. Ron Fricken outside or as we move out. But just some follow-up items. We do have a One Health Academy survey, so we want to continue to make this experience meaningful for you guys. We'll have that open until tonight. And just please encourage you to access that via the emails that we've sent out to share some insight if you feel like we're missing anything or you would like to be heard. And finally, we hope everyone has a great holiday or had a great holiday. Enjoy any time off that you're able to, and we will start back in the spring on the second Wednesday of the month from January until June, so that will be a consistent date for us here at ASM. Thank you so much to ASM for hosting us, for One Health Commission for providing our webinar, and thanks so much for coming again, you guys.